And without further ado, let's see. Okay. So we're going to talk about, so I just added this slide last minute. And the focus of today is finishing molecular biology, finishing the central dogma, as we call it, of how a genetic code is read in your DNA. And from that DNA genetic instructions, a protein is ultimately produced. But I haven't really defined for you what a gene is. I may have mentioned I always am updating my lectures. I'm always changing them, always improving them. I'm striving to be the best I can be. So I just added this slide like 30 minutes ago. So you won't see it, but I'll just give you some time to write it down, what I have here that I added. But a gene is a region of DNA. It's a stretch of DNA in your genome, which is the entirety of your DNA in the nucleus of your cells. And it's a specific region that will code for a specific protein. So you'll have a gene for the amylase digestive enzyme. You'll have a gene for keratin, the structural protein. You have a gene for producing antibodies in your white blood cells to fight infection. And, these are other proteins we've talked about before. You have a gene for each one, a different gene for each one, a different region of DNA in your 23 chromosome pairs. Remember, they're homologous, so there's 23 chromosomes of information. So if you have a gene for keratin on your chromosome 8, you have another gene for keratin on your other chromosome 8. Okay? Weird, crazy. I know. But yes, so that's what a gene is. And today, the focus, contractile proteins? Okay, yeah, so they're just actin and myosin. They're these filaments that they slide past each other when a muscle contracts and relaxes and extends. That's all. No worries. Okay. But the main focus of today is the how. So I talked about last time that I love this analogy. I actually went and Googled, I couldn't think of like examples of recipes from a cookbook. So I Googled some. There's a lot of soup recipes out there, a lot of potato salads. But anyways, I like this example of basically, maybe it was my other school I talked about it. No, I think I did talk about it here. I did. Okay. But I do like this analogy of the cookbook where DNA is your cookbook and the gene is a specific recipe. Okay. And the cell is going to read that gene and read the instructions on how to make your protein, which would be your pizza, your soup, your potato salad, whatever it is. And the kitchen where you're making your protein is the ribosome. That's where protein synthesis occurs. We learned about that a few weeks back when we talked about organelles. But today, like I said, we're going to be focusing on how this happens, how we read the gene, how the cell will read the DNA and interpret it, decode it to make that specific protein in the kitchen, in the ribosome of the cell. So, okay, let's do it. This process I mentioned, we call it the central dogma of biology. Okay. Because it really is just the most fundamental, I don't want to say important, but maybe the most critical process in the cell. It's so profoundly important that this is occurring in our cells. It's how our cells do their job. So we call it the central dogma. And so let me okay. The central dogma is this. DNA is what we start with. DNA is read to produce RNA, which is a different type of nucleic acid. We're going to talk about these molecules in detail. That's what the chart is on the slide. And the RNA is read and decoded to produce the protein. So the RNA is sort of this intermediate molecule in this process. But the central dogma is going from DNA to RNA to protein. The information flows in this way. DNA is read to produce an RNA molecule. The RNA molecule is then read to produce a protein. Okay. In our central dogma, in this central dogma, the process of converting the DNA, you're not converting it, you're reading it to produce an RNA molecule. That process is called transcription. It's our first process of this central dogma. So DNA is transcribed into RNA. So transcription is that first step. Okay. Now, the process where RNA is used to make protein is called translation. So RNA is translated into protein. So before we get into the specifics of transcription and translation, I want to talk about these molecules: DNA, RNA, and protein. And I want to talk about, so I'm actually going to, let me see, I want as much room, I think I can fit here, but I'm going to rewrite them. So DNA, RNA, protein. So we're going to detail out their characteristics here. The first thing we're going to look at is their structure. What are they? What are they made of? So it's sort of a chart. So if you look at the slide, this is what we're going to be filling in. So the first row is going to be the structure of the molecule. The second row is going to be the component, or the components, what it's made of. What is it made of? The reason I'm doing this is because there are some differences I want you to be aware of. And I think it'll help you also reinforce the idea that DNA and RNA are nucleic acids and proteins are an entirely different class of molecule. But there are some other subtle differences you need to know. The last thing I want to talk about is function. We've talked a little bit about function of DNA and protein, of course, and I just want to go into a little more detail now. So DNA, RNA, and protein. DNA is double-stranded or single-stranded? It's a double-stranded, double helix, right? Nucleic acid. So it's a double-stranded nucleic acid. And RNA, in contrast, is single-stranded. It's also a nucleic acid, so it's a single-stranded nucleic acid. And there are a few types of RNA molecules, but we'll be looking at that shortly. I just want to show you, though, it's still a nucleic acid polymer, and this is what it looks like. So notice how the DNA, of course, has the two strands, complementary strands. It's double-stranded, and then RNA just is a single string or single chain of these nucleotides, right? So that's one key difference between DNA and RNA. And then, of course, proteins aren't nucleic acids at all. They are three-dimensional polypeptides. And on the slide, as you saw, it's the ribbon confetti-looking three-dimensional structure. Okay. What's it made of? What's it made of? DNA is composed of, we know, nucleotides, right? But I'm going to sort of shorten that for lack of space. And I'm going to write the 4 different types of nucleotides. And remember, the 4 different types of nucleotides all depended on the base, right? The nitrogenous base. You could add adenine, thymine, cytosine, or guanine. We talked about this last time. So DNA, you can have an A, a T, a C, or a G as your nucleotide. You've got 4 choices. RNA, also 4 choices, 4 base possibilities. But the difference that I want you to know is that there's no thymine in RNA. RNA does not contain any thymine bases. It contains a different one that's similar looking, but it has a different name. Yeah, Sana, it's a U. That's correct. So it's uracil. So RNA has adenine still. It does not have thymine. Has adenine, it has cytosine and guanine, but it has uracil, which is a U. So this is uracil. And if you look at the sequence of nucleotide base pair or bases here, you'll notice no T's in the RNA, right? And the uracil is in place of the thymines. So you have U's instead. And then proteins are composed of amino acids, right? Of which there are 20 varieties. You learned that in the beginning of the semester in the first unit. Okay. Function. DNA, we've talked about the function of DNA. DNA's function really is storage of genetic information. It's the cookbook. It has all the instructions for our proteins. So storage of genetic information. Okay, RNA, we got a bit to write. So I'm going to kind of squeeze it in here and I'll read it out to you. But there are two types of RNA that we're going to talk about in this class that play an important role in this central dogma. The first one is mRNA. mRNA stands for messenger RNA. So this is messenger RNA. Move this up for you. And messenger RNA acts as a message of a DNA sequence, so specifically a gene, during transcription. Since we can't really write all that, I'm going to write, or we can't fit it here. And we're going to look at this in detail on the next slide, but I'm going to write RNA complement of DNA sequence. And we're going to see that its function as this RNA complement, it's going to be important in transcription. In fact, why don't I put a red number 1 here? Just to show you that mRNA's main importance is involved in transcription. And then the next form of RNA, tRNA, is its main importance is its involvement in translation. But I'll put it down here actually, why not? So the next form of RNA is tRNA, but it doesn't stand for translation RNA, it stands for transfer RNA. Transfer RNA. And like I said, it's going to be involved in translation, which we'll take a look at. And it's going to be involved in the transfer of amino acids based on mRNA sequence. Now, of course, this doesn't make sense to you yet, but this is one of those, I like to do these tables as a, to have it in the back of your head for when we look at it in detail, but also when you're studying, you can refer back to it as a study tool. Because we're going to look at this in detail soon. Okay, sort of bled over here into the protein column, but we've talked about the function of proteins before. There's many, right? Many functions. Who remembers some? Let's write some examples. I actually mentioned a bunch of proteins on that previous slide. So what's an example of a protein and its function? Building stuff. That's true. I can't. So there are synthetases involved in building, but we didn't talk about that too much. We talked more about structural proteins. So let's do that first. Any other examples? Structural. So like keratin. Movement, absolutely. Muscle proteins. Enzymes, catalyze chemical reactions, totally. And I ran out of space. Oh, fight infection, totally. I ran out of space, but that's, good enough. Good enough. Just to know that there's many functions and we've talked about them. Okay, cool. All right, so any questions about these molecules and their characteristics before we look at transcription? Okay, cool. Does anyone watch the World Series yesterday? You're in Orange County, so you guys like the Angels? Maybe, I don't know. Angels, or are you Dodgers fans? Dodgers, interesting. Cool. Yeah, I think San Diegans were supposed to hate the Dodgers because, I don't know, I think we're just jealous because we're not good, the Padres. All my friends are like, nah, I'm so angry, the Dodgers. And I'm just like, because they're good? So they're like, they're way better than us? San Diegans, I know. I pretend to like baseball too. I kind of, when it comes to sports, I think it's the only thing my brother and I have in common, that we're not super into sports. But I don't know, if there's like a championship, basketball, finals, World Series, I tend to enjoy watching those, but otherwise I'm not too into it. Okay, so let's take a look at transcription. Transcription process. Okay, so during transcription, as you can see here, as I have the template, pun intended, we're going to be looking at a DNA template. DNA and mRNA. It's this process of reading DNA and producing an mRNA molecule from DNA instructions. We're going to, and I'm going to go over with how they do that, how this enzyme will do that. But first, let's define it. What are we doing here? This is where the cell makes an RNA, specifically an mRNA, messenger RNA, mRNA transcript, which is just the RNA molecule. It's being assembled of a specific gene, which we know is in the DNA, right? See, I think I'll have time. Let me see. Okay, so let me also, just like as an example, the insulin gene. So insulin is a protein. It's a protein hormone. Hormones are signaling molecules that are secreted from cells to have some kind of effect somewhere else in the body. So insulin is this protein that's secreted by the pancreas to regulate blood sugar. Essentially what insulin does after you eat, the pancreas spews out a bunch of insulin, it circulates in the blood, gets to other cells, and tells these cells to take in sugar from the blood, glucose actually. It's how glucose gets into cells. It's insulin starts the whole process. So just as an example, we'll use insulin. Well, yeah. But it could be any other of those protein genes for these proteins, right? Keratin, amylase, digestive enzyme, etc. All right. So the slide, we're starting with DNA, right? So let's say we're looking at the gene for insulin and the specific DNA sequences as such. I'm going to write it here. Okay. That's our DNA sequence. Say it's our gene for insulin located, you know, on, we have 3 billion base pairs of DNA along our 23 chromosomes. So genes actually are not this short. Let me just say it for you. They're at the minimum, they're going to be a few thousand DNA nucleotides long for a gene. But obviously, for simplicity, we'll look at this. Okay. So the cell is going to transcribe this gene into mRNA. It's going to read this DNA template and assemble nucleotides of mRNA and create this mRNA molecule. Okay. I'll show you how it does that. I mentioned that we talked about last time, there's 5' end and 3' end, the anti-parallel nature of DNA running in opposite directions. And this is pretty important because there's the enzyme in transcription, also in DNA replication, is these enzymes, these polymerase enzymes, right? Remember DNA polymerase from DNA replication? Well, the enzyme involved in transcription is called RNA polymerase. RNA polymerase. RNA polymerase. We're going to assemble an RNA polymer, right? So let me put mRNA here because that's what we're going to be doing. But what I was saying is that these polymerase enzymes read DNA from 3 to 5. That's just how they work. They read 3 to 5. They synthesize 5 to 3. 5' to 3'. So we call the 3 to 5' strand on the DNA the template strand because our polymerase enzyme during this transcription process, well, what happens, there's a bunch of enzymes involved, but the DNA double helix is going to come apart more like this, right? It's connected by those hydrogen bonds and it's going to come apart at this gene. Let's say we needed insulin. It's going to come apart a little bit. This RNA polymerase is going to sneak in there and start synthesizing this mRNA single-stranded molecule. How does it do this? Well, just like in DNA replication, except, of course, DNA replication was both strands being synthesized, right, to create another double-stranded DNA molecule. It's just going to synthesize one strand, reading this template by adding in complementary bases to that template strand. So we haven't practiced this yet, I don't think. But what is the complementary base to T, thymine? It is. So we talked about this, right? A. A always goes with T. So this polymerase enzyme is going to throw in an A to create this mRNA. Okay. What's complementary to T? Or I'm sorry, to A? What will come next? Ah, I got you guys. It's not T, it's U. Yes, yes, we're RNA. There's no thymine, remember? Right. Okay. What comes after that? Complementary to C. G, yes. And then I'll just fill in the next part. So this is U, C, U, A, C, C, A, and then that's all we got. That's our mRNA strand here, mRNA sequence. That's complementary to this template strand on the DNA. The enzyme read it 3 to 5, synthesizes this molecule 5 to 3. But okay, so question. Okay, so normally in DNA, the complementary base pair to thymine, or I'm sorry, to adenine is thymine, right? A pairs with T every time. So you got A and T, TNA, 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 AT. But there's no thymines in RNA. There's only uracil. Uracil binds complementary to adenine. No thymine. There's no T's. So if you want to put a T complementary to this A, right? If you're an RNA molecule, there is no T. So you put a U instead. It's a uracil nucleotide. So you only have U's in RNA when we're talking about RNA. Okay. Wow, my voice cracked. That was embarrassing. Okay. Jeez. See, I learned being a professor, you talk a lot, and I think my voice is, and it's dry out. Okay. Promise you my voice hasn't cracked in that long. Jeez. Anyway. Okay. We've got our mRNA sequence, we've got our molecule, and let's take a look at it. The next thing I want you to know, well, transcription is done. We've transcribed the DNA into mRNA. Every 3 nucleotides of this mRNA, we call it a codon. So this is one codon. This is another codon. And then this next 3 is another codon. Of course, this would continue on. Usually these are thousands of nucleotides long. Here we have 3 codons shown. So one codon has 3 RNA nucleotides in it. This is going to be important in translation. Okay, let me ask you guys a question. If we're starting with DNA, where does transcription happen in a eukaryotic organism? If we're starting and working with our DNA here, where is this going to be occurring in the cell? In a eukaryote. Where's our DNA? Yeah, the nucleus. Perfect. So we're starting at the nucleus. Absolutely. I say eukaryote because, of course, prokaryotes also do this. Bacteria do this. It's essential for life. All life on this planet does this process of transcription and translation, but they don't have a nucleus. Their DNA is just floating around, so they do it in their cytoplasm. So just keep that in the back of your mind. If we have time, we're going to do the whole process and take a look from the nucleus to our next destination, which is the ribosome. Oh, never mind. We're going to talk about tRNA first. Okay, let's do that. Talk about tRNA first. Okay. So just really quick note about transfer RNA, and we'll do a little drawing. Okay. So we looked at codons in messenger RNA, right? That's going to be important soon. But I want you to take a look at a tRNA molecule. This is a tRNA molecule. This is like the three-dimensional form. Same thing. But at one end of the tRNA, you're going to have an amino acid attached to it, okay? At the other end, there's 3 nucleotides in the tRNA molecule that are the anti-codon. So codons we find in messenger RNA. Anti-codons we find in tRNA. This is going to be important in translation. Remember, tRNA is involved in translation. So we're going to talk about that next, but let's just draw the tRNA just so you have it in your notes. tRNA are transfer RNA, right? Transfer RNA. Kind of looks like a T, funny enough, when you unwind it. Looks like this. And there's going to be an amino acid attached to the top of it. And this bottom part, let's say it's ACU. That's our anticodon. That's what tRNA looks like. And there are many tRNAs, and they all have a specific anticodon 3-letter combination or sequence with a specific amino acid to that sequence. And we're going to take a look at, eventually we're going to look at what I kind of mean by that. And the sort of, depending on what your anticodon is, well, that will determine your codon. We'll see that shortly. And depending on your codon, what amino acid will be attached to the tRNA associated with it. That probably makes no sense right now, but we're gonna look at this. I just want you to know what this tRNA looks like. It has an anticodon region with a specific amino acid attached to it. Okay, so now let's take a look at translation. Translation. All right. Now, transcription occurred in the nucleus. We assemble this mRNA messenger RNA molecule. The mRNA molecule, this message, is going to leave the nucleus through a nuclear pore in the nuclear membrane. It's just going to be able to leave it, and it's going to go to a certain organelle involved in protein production, the kitchen, the ribosome. And this mRNA is going to be read and translated into protein. This is where the cooking happens, okay? So translation process. Like I said, we're at the ribosome. So we've left the nucleus. Remember, ribosomes, there are free-floating ribosomes. There's also ribosomes on the rough endoplasmic reticulum. And they kind of look like this. They kind of look like hamburgers. And there's a small ribosomal subunit and a large ribosomal subunit. Okay. Yeah, subunit. Yeah. What's going to happen is the mRNA that was produced in the nucleus and leaving the nucleus, it's going to find a ribosome and dock onto the ribosome and actually thread through it. So this is going to represent my mRNA strand. This is my mRNA. And let's say, well, I'm going to actually use the mRNA we produced through transcription. So it came out to be, oops, AUG, UCU, ACCA, right? That was our mRNA strand. What is going to happen here? We're going to have tRNA molecules come in at the ribosome. And remember, these were our codons, right? Oops. So every triplet codon, triplet nucleotide codon of our mRNA actually has a corresponding anticodon tRNA. The anticodon for, let's say, this AUG codon, I'll just write, this is codon, right? These are codons. The anticodon to this AUG codon is complementary to it. The same base pairing rules. So to AUG, that would be a tRNA would come in, the specific tRNA with the anticodon on it, which would be U, A, C. Complementary to that codon. This is the anticodon. And so you would have a different tRNA match up with this codon. Of course, this is drawn not to scale, really bad. Let me draw this one better. I'll try to at least. Okay. So you're going to have these specific tRNAs come in with amino acids attached to each one. And I'm actually going to write specific amino acid. You're going to see on our next slide that depending on what the codon sequence is, what combination of 3 letters, it's going to determine which tRNA and which specific amino acid with the complementary anticodon is going to come into the growing protein that we're making. Oh, I just kind of saved time by not doing the anticodons, but it would be complementary, right? So A, G, A, U, G, G. Okay, and so the next thing that's going to happen is a bond will form between these amino acids. This is a protein. Proteins are amino acids with what we call peptide bonds formed between them. That's just the fancy word for the covalent bond between amino acids. So what this tRNA is doing in translation, tRNA matches each mRNA codon and transfers in a specific amino acid to the growing polypeptide, or the growing amino acid chain. Polypeptide chain slash amino acid chain. This is how you make proteins. Okay. Any questions? Any questions? Okay, cool. So this is how insulin protein would be made. Okay. So I kind of alluded to how codons are recognized by their specific, or I'm sorry, I should say, how we can see what amino acid this codon will eventually code for, with which specific tRNA with a specific amino acid for this codon. So we actually call that the genetic code. Oh, just kidding. We're not there yet. That's the next one. Let's do a discussion question. Way to go, Ryan. Should add that in my complete notes so I know for next time. But take a look at this question. Take a minute or two and answer how many amino acids a gene with 45 nucleotides will be produced. How many amino acids will result from a gene with 45 nucleotides? Itself, that's correct. Yes. Cool. So it is 15. And the reason is because if you have, so Joey, that's exactly right. Okay. So to summarize what Joey is saying here, if you have 45 nucleotides in your DNA of your gene, right? And they are all transcribed into mRNA, and you get an mRNA strand that has 45 of these nucleotides. Well, each triplet nucleotide is going to be one codon, which gives rise to one amino acid, right? So if you divide that number by 3, you get the amount of the amino acids. If you have 45 nucleotides, for every 3, you get 1 amino acid transferred in during translation. So you'll get one from 45 nucleotides, you get 15 total amino acids. Exactly. All right, cool. So again, every 3 nucleotides of the mRNA sequence codes for 1 amino acid. And so we're going to actually take a look now at the genetic code, which is this amazing thing. And I'll tell you why. But it tells us for a specific combination, every type of combination you can have of for a codon of mRNA nucleotides, what specific amino acid will be transferred in during translation based on that codon. And here it is on the right side there. That's the genetic code table. So take a look at that. We'll erase the board here. Okay. Let's write the definition here down, and then we'll go over it. You're way ahead of the game. Yeah. Methionine is the first amino acid for every protein. Yeah, we call it the start codon, and it's the first amino acid. Yeah. I'll show you guys how she determined that. But let's define the genetic code. What is it used for? It's used to determine which amino acid, remember there's 20, right? 20 different ones, is coded for by a specific mRNA codon. DNA song Bruno Mars remix. Is it biology related? I think maybe I've heard of what you're talking about. I forget. Bruno Mars does a biology thing? Really? That's cool. I'll check it out. Oh, it's a remix of his song. Oh. Yeah, I'll check it out. Sweet. Play it in class in the future. Oh. Sick. I'll write that down. I'll check it out. Oh, Joey, thank you. It's like, I'm going to bookmark it. Oh, it has a lot of likes. Sick. Thank you. I'm going to play it in class in the future. I think that'll be a big hit. Okay. All right, so let's take a look at the table. Wherever it may be. Okay. So this is how you use it. For a codon, right? Triplet nucleotides of mRNA. So you have a first letter for it. Then depending on your second letter, if it's U, C, A, or G, you go up here. And then your third letter on this side. So we're going to do an example of this. But I also want to point out that these 3-letter abbreviations are for amino acids. So leucine, serine, threonine, asparagine, serine, and this methionine, AUG, it's in green. It's known as the start codon because every protein starts with AUG and a methionine amino acid. So we're going to do an example of using this table. So if I told you that the mRNA sequence is, mRNA produced during transcription is CUC AUC, GCU. I'm going to put up the, so write that down as CUC, AUC, GCU. I'm going to put up the table, and then I want you to tell me the amino acid sequence produced during translation. So the amino acid sequence. Okay, take a look at the table. I'll give you a minute to tell me what amino acids I'm going to write. And you can just tell me the 3-letter abbreviations. Not methionine. I don't have an AUG, right? CUC, AUC, and GCU. Joey's got it. Joey's got it. He's quick. He's quick. All right. So for that first one, CUC, right? C, first letter, second letter, U. So we're here. And then the third letter is C, so it's up here. Well, all of these code for leucine, right? LEU. That's how you use the table. So I'm just going to go ahead and write the sequence. It is LEU bonded to ILE bonded to ALA. Okay. So 2 important notes about the genetic code, and then well, probably just end early. Might just do the other thing next time, where I won't draw it all out. I think that's a good call. Don't say yay for ending early. Biology's amazing. Hello. Don't you want to be in this class forever? Like, don't you wish we could be talking about this all night? Probably not on Zoom or in person. I mean, I'm joking. I'm totally joking. All right, so let's talk about 2 more important notes about the genetic code, one of which you've already seen. The genetic code is redundant. So what this means is that multiple codons code for the same amino acid. AA amino acid. Kyle. That's funny. Okay, check it out. So take a look at the table here. You'll notice there's this redundancy where if we looked at the, let's just look at this, for example, UCU, UCC, UCA, UCG, those all will code for serine. Those all will have a transfer RNA with serine attached to it. So you'll see this redundancy, right? There's multiple codons for a single amino acid. And then one more important note that I think is pretty remarkable is that the genetic code is universal. This means that every organism on this planet, all life, single-celled, multi-cell, prokaryote, eukaryote, fungi, bacteria, human, elephant, you freaking name it, jellyfish, plant, every organism uses this code, uses the same code, the same chart. If you have this mRNA sequence produced in an ant, an insect, a spider, in a lobster, in a sea star, it's going to make this same amino acid sequence. So I think that's pretty amazing. It kind of demonstrates this common ancestor evolution theme where all life is related in this way. We all have this same machinery to produce proteins. We don't all make the same proteins, but we all read mRNA in the same way. All right, so 647, it's really all I wanted to talk about. Lab questions. So first, let me answer any questions about the lecture, and then I can answer labs as well. You have lab questions. Oh, goody. Let's talk about it. But any lecture questions? What does the table mean when it says stop? Great question, Cole. Thank you. That's an awesome question. I didn't talk about that. So stop is a stop codon. So at the end of every protein, there's a codon, a stop codon, and this actually terminates translation. No amino acid will be transferred in, and the final protein product will fold, and you'll get it will stop translation. So at the end of every mRNA sequence, you're going to have one of these stop codons. Great question. Yeah, that'll cause it to just fall off the ribosome, and you get your final protein. Keep people on. No, people can leave. Plus, it's not even 6:50 yet. Is there stuff to cover on mutations? No, that's a great question. We're going to be talking about mutation next class. Because we're going to do, yeah, I think so let me take a look. PowerPoint. Yes. So next time we're going to be looking at we're going to be doing another practice problem, just like a warm up. And I'll talk about mutation then. It's already there. Reminding myself. Okay. I'm back. Here we go. Okay.